Okay, everyone, it's that time. Let's get started. Let's see, announcements. The lab. Has everyone got their Scarlet by Brown crosses set up? Has anybody not got it? I'm kind of impressed. Yeah, most years there's always a couple of stragglers who take a while to get their versions set up, and you guys just are racing ahead. All right, what does that mean? That means this week we're just kind of watching things, waiting for them to grow. If you've got a model with lots of larvae in it, that is the Scarlet by Brown cross, or if it's been 4 to 6 days, somewhere around there, that you've had the flies in there, then it's time to clear the flies. So get rid of the adults. So again, what we're expecting is next week you're going to come into the lab and we'll start finding all of our wild type F1 progeny. We expect wild type F1 progenies. So we're going to be looking for that next week. That's what we're going to be waiting for. Next week in lab, what we're going to be doing is some fun stuff. We're going to learn how to wash glassware. Yeah, aren't you excited? Yeah, we'll show you how to clean up the fly bottles. But also we're going to talk about databases and where you can find information about genes. And also, and this is kind of important, remember you're supposed to write a lab report. Actually, you're going to write 2 lab reports. I'm going to get you started on that next week. So lots to do. Okay. The other thing is let's see, let me think. The exam. Oh yes, the exam. You probably noticed I finished the exam. There is an answer key posted with the old exam. You can go browse through that. One important thing is I was checking everyone's results, and Canvas was automatically misgrading problem 12. So I have corrected that on everybody, but it tells me you might want to double check. And if you find something different from here, I tell you in the answer key that this is the correct answer, and you say, I got that answer, but Canvas counted it wrong. Let me know so I can correct it. Another important thing from the exam, remember, one question was just asking you to tell me what you think of the course so far, all that kind of stuff. A lot of people said they wanted more homework. You weirdos. I mean, this is typical UMM student stuff. You guys are always so industrious. But anyway, so what I've been thinking is every week I've been giving you this homework assignment, except for last week, worth about 10 points. Would anyone object if I made it 20 points each week and threw in more problems? You would object. Yes, go ahead.
Could you have more than one attempt?
Could you have more than one attempt? Ooh, that's an idea. I'll see what I can do about that.
Objection.
Yeah.
Because then we can come to your office hours and see what we're doing wrong, and then get full credit for these.
Sure. No, I will tinker with Canvas and see how I can make it dance. And I'll see if I can make it so, yeah, you guys get multiple tries at it. That's fine. Because the main purpose of those exercises is to force you to go through the problems. And if I make you go through it twice, that's making you think even more. So I got no complaints there. Okay, I'll see what I can do. So that nobody has objections if I increase the point value of the weekly exercises to 20 points, which is going to weight them a little more heavily in the final grade. So, but I don't think anyone's afraid of that. And we'll see how it goes. And I'll be posting some more later this afternoon, so I can ruin your weekend. Okay, here's where we are. So you can see from my list up there, I got a whole bunch of things I packed into this lecture. And looking at it afterwards, I was thinking, Man, that's a lot of terminology and a lot of words. So I will reiterate again that little phrase I always put up there. Ask questions. Go ahead, slow me down. Yeah, there were a few comments about, well, my pace is okay, but sometimes a little fast. Yeah, go ahead. Don't be shy. You can tell me to explain things twice, just like I make you do problems twice from now on. Anyway, so here's all the things we're going to talk about. And I want to start by just mentioning this. So we've been talking about Gregor Mendel and his model for how genetics works. It's a fairly basic model. And this is kind of a relevant point here. It was published in 1865. Mendel and Darwin really were contemporaries because Origin of Species was published in 1859. Mendel comes along a few years later with this in 1865. Nobody read it. So this is the paper. Research on plant hybrids. You know, if I had a time machine and could go talk to Gregor, I would tell him, you got to work on your PR angle a little bit, okay? Because this is not a catchy title. That's a really boring title. And the paper itself is kind of drags. It's a lot of numbers, table after table of numbers. And so, like I say here, it got ignored, horribly ignored for about 35 years because nobody read it. You know, contrast that with Charles Darwin. He was an eager correspondent with scientists all around the world. He was promoting his book, you know, in writing. He didn't travel much, but he's promoting his book. Everyone knows about it. He's got a publisher who makes a big deal about it. So just for you who go on to scientific careers, don't do that. You know, we get these biocommunications courses here. I can't emphasize how important those are for your academic career. You want to learn how to do this stuff. Because otherwise, you're like poor old Mendel. You publish this stunning paper that's going to revolutionize all of the study of heredity, and it's ignored for 35 years. How terrible. What a waste. Anyway, but it is rediscovered in 1900. That was good. There's a bunch of scientists who simultaneously rediscover his paper when they're browsing through the literature, looking at all the back papers and seeing what's cool. 3 of them at once find this independently, and it makes a big splash. But it took that long, and he was dead at that time. Isn't that tragic? Yeah. If any of you discover something marvelous, make sure everyone knows before you die. That's a good idea. Okay, so it's rediscovered in 1900, and here's the sad thing. It got really grossly abused for a while. Those of you who know your history may know that this period from 1900 to about 1940 was a hotbed of eugenics. People read Mendel's paper, and although Mendel's paper said nothing about this, said nothing about races, said nothing about uses of genetics for improving the species or anything like that, it became a foundation for arguing that racism had a scientific basis. So like here, this is this weird poem published in the 1920s by this guy Desjardins, and somewhere here there's a paragraph here. This is the law of Mendel. No, the stuff he's talking about is not the law of Mendel, right? So he's just arguing for defectives will breed defectives. Why do we allow these people to breed back to the monkey's nest? We should only breed the best. Yeah, this is eugenics. This guy, Desjardins, actually testified in the Buck versus Bell case, which Some of you may have heard about it. It was an important stepstone in the eugenics movement, where they obtained Supreme Court approval to sterilize people against their will if they weren't up to their standards. Okay, so we won't go into detail in the history of this. It's a sad and sorry history. Okay, so it was a real problem. It culminated in the Nazi atrocities in the 1930s and 40s. We're trying hard to pretend it never happened, and a lot of people now are coming back to a new eugenics, new racialist views. Maybe somewhere in here by the end of the semester, I can say a little bit more about that. But let's move on, because the problem with this kind of interpretation is that these are people who really don't understand genetics. They have a cartoon kindergarten version of genetics. The stuff we've been talking about. You know, hey, genes, you have dominant or you have recessive, that's it. And so what we're going to start talking about today is how that's not always true. Genetics is a lot more complicated than that. That there's a lot of other details that we can overlook. So one of the things I want to talk about first is, here's an appropriate use of genetics. This is generating pedigrees. This is a really common thing to do. Genetic counseling, one of the things they will do with you, if you go in with a possible hereditary defect, or you know somebody who is to do the analysis. They'll go digging into your family tree. And they're going to be looking for where this problem might have come from, or whether it's not really genetic at all. So here's an example of a simple pedigree, illustrating a couple of features here. So this line here, that's called a sibship line. This is marriage. Squares are males and circles are females. You can also see down here a couple of keys. There's identical twins, that little triangle there, and there's fraternal twins. So we can illustrate all this stuff in this kind of diagram. The propositus or proband, it's called one of those two things, is simply the person that triggered the study of the pedigree. So this could be like, if I go in for genetic counseling and I say, hey, I think I'm really kind of funny looking. Could you look at my family tree and see if this is hereditary? Yeah, my brother's also kind of funny looking, so it must run in the family. That would make me the propositus here. So that's all that means, is this is the person who called the whole thing into attention. Okay, so we will sometimes use pedigrees to analyze the inheritance of a trait. Okay. Here's a few terms to know. Yeah, like I said, this is female, this is male. Put a diamond if you don't know the sex. Affected individuals are sometimes, in some pedigrees, they're colored in. So that's what we see here. There's the parents. They are presumably unrelated. That's what the single line means. Sometimes you have consanguineous parents. That just means they're related. So if you marry your cousin, your pedigree will have something like this. Or worse yet, if you marry your sister or brother, no, you don't want to do that. You will get this kind of marker there. And it's kind of an alert that, hey, there's some inbreeding that could be going on here. So take that into account in your analysis. And there's our offspring listed there. Sometimes they will list the birth order, not always. There's our fraternal and identical twins. Sometimes in complicated pedigrees, you'll find things like this, where they just put a number there. Say, okay, this person, they had 4 boy children and 4 girl children. And they're all unaffected. That's all we care about here. Yes, sometimes you'll see deceased individuals, and sometimes we'll talk about heterozygous carriers. The other way, this is off, see the little dot in there? That's if you do everything in black and white, but as you can see here, sometimes we'll do this. Do a half and half coloring of the individual. So that's basically all you need to know to read a pedigree. Now, here, for example, is a pedigree for a rare recessive trait. When analyzing a pedigree, one of the things you want to know, if you suspect a particular trait to be inherited, you want to know, is this something really common or something really rare? Because that will affect how you interpret these marriages here and other marriages down here. If it's a rare trait, you will generally say, I assume that anyone who is not related to you that you've married from the general population does not carry the trait. So we'd say they're homozygous for the wild type trait. On the other hand, when we look through a pedigree like this one, we start tracing everything back. Okay, here we got some individuals who are clearly affected. We got one up here. So these people with this girl and this boy would say, hey, my mom and dad did not have this trait, but grandpa did, right? And so they'd be asking, okay, what's the probability of passing this along? What we really want to know is, what is the pattern of inheritance of this trait that I have? You know, if we see it skipping a generation like this, where it does not appear here, but it is here, that sort of suggests, okay, it's probably recessive. As you might guess, you'd then say these individuals are probably homozygous for the trait. And if they're coming to you for counseling, you'd say, okay, you're a carrier. Your children probably will not express the trait, right? Because you're going to marry somebody outside your family, I hope. And then what you'll get is children who, 50% of your kids will be heterozygous. And the other 50% will carry only the dominant wild-type allele. So that's the kind of information people like this would be looking for, is trying to get advice on having children. Yes?
Quick question. What does that diamond symbol mean?
The diamond symbol means they don't know.
Oh.
Yeah, they don't know the sex of that individual. Sometimes that's because it's a child who died in infancy. It's because this was so long ago, we don't know anymore. Birth records are inadequate, that kind of thing. Which brings up another point I should mention. Often when you kind of go to get these pedigrees done, you'll find that they're generally about 3 generations long, about 3 generations deep. So yeah, I kind of expect most of you remember your grandparents, but not all, of course. But few of you might remember your great-grandparents. And if you do, you don't remember much detail, except they were really old, right? And great-great-grandparents, they were all dead, so you've forgotten them. So there's probably a 3-generation deep window that we're looking at. You can, of course, go to official records and see if there's a record somewhere of some heritable defect in the family. And for that, you need really good genealogy tools. I used to live in Salt Lake City, and it was kind of a blast to go down to the archives there because they record everything. And you go digging around, and yeah, I could find my relatives back like 5, 6, 7 generations. I'd have to be pretty diligent to go back farther than that, but in some of the records you could. So three generation window is typically what we see in this kind of thing. Okay, just to revisit a problem. So we ask, what is the probability that these individuals, so this is 1 generation, 2 generation, 3 generations, so 2, 5, for instance. You know, what if this person were asking, what's the possibility that I am a carrier? What would you say? So it's a recessive trait.
50-50.
Probably not. So this person has a sister who is a carrier, so we know that. So that means that we know that one of the parents has to carry the allele, right? What's the probability of that, though? We got to multiply things here. So what's the probability that one of the parents is a carrier, knowing that one of their children is also a carrier? Here's going to get a little bit dicey, because we know for sure that one of them has that little a allele. So the question is, what's the probability that any one individual here will inherit that? Now in this case you would say, okay, let's see, if it's 1, and let's just assume it's one of the parents that has it, because this is a rare trait, okay? But it's also possible that they're big A rather than big A, little a. So if we got one parent here who's got it, the probability that any one of their children will inherit the little a allele is, yes? 25%? Yeah, well, no, actually, we have to assign a probability to whether they're big A, little a or not. It's going to get hairy, but whatever that probability is, let's say it is 25%, so there's 25% it would be passed on to this individual. Then we ask, okay, what's the probability that this individual is the carrier? Because there's other kids here. This one's got it. We look here, we see 1 in 4 has got it. So just assuming, let's for a moment, just assume that one of those parents has big A, little a genotype. And then we ask, what's going to happen here? Well, this individual is it's, again, 50% chance that they've inherited it from the one parent. So we'd say 50% here. If both parents carry it, it gets messier. So we won't get that. We want to know a lot more about this family history. So if this person came in for genetic counseling and said, okay, my sister had some children with this particular heritable trait, you'd say, okay, we got to look at your parents who are not affected, and we got to look at their grandparents to figure out the probability. Genetic counseling gets complicated. Okay, here's an example of complicated pedigree. Oh, look at that. There's lines all over the place. This is because we have to sometimes deal with things like somebody gets married 3 times. So here's the person we're interested in. This is Woody Guthrie. Who's heard of Woody Guthrie? You have, okay, a few of you have. Really famous folk singer. Without Woody Guthrie, we wouldn't have Bruce Springsteen, we wouldn't have Johnny Cash, we wouldn't have all these people. But so he's a really influential. He did the song "This Land Is Your Land." I'm sure many of you have heard of that. Anyway, he was a very famous, very popular folk singer. And yeah, he got married 3 times. Tragedies in his family, for instance, you notice one of his children died in a car accident. Here's another one who died in a car accident. The lesson from this is you don't want to drive anywhere, right? Stay out of those cars, they're dangerous. What's most interesting about this, though, is Woody Guthrie had a disease. He had Huntington's disease. Huntington's disease is caused by a dominant allele. So previous diagram, that was a recessive trait. Now it got passed on. This is a dominant allele. It's a particularly terrifying disease because Huntington's disease has late onset. So you're living through your childhood happily. You get into young adulthood, you're just fine. You're becoming famous singing folk songs or whatever you do for a living. You get married multiple times, you have multiple children, and then in your 40s you suddenly discover that your brain is going. So this is a progressive deterioration of the brain. And the symptoms are you gradually lose motor control, you become weak, you exhibit instability, emotional instability, lots of problems like that, and then you die. And because you had that happy childhood and you had lots of kids, they get to watch you die. Oh boy. Furthermore, because it's a dominant allele, your kids get to watch you die and think, oh, I've got a 50% chance of inheriting that same disease and of it hitting me in my 30s or 40s or 50s. Now, doesn't that sound horrible? He had, like I said, he had a bunch of children. Again, some of you may have heard of this one, Arlo Guthrie. Who knows Arlo Guthrie? Musician. You know, Alice's Restaurant, that song? Oh, man, this is a liberal arts college, so I can tell you, go look up Alice's Restaurant. It's a fun song. Anyway, he's famous for that. So he's another folk singer. And imagine the dilemma he's got. Is he going to die of this horrible disease in the near future? Is he going to pass it on to his own children? The happy news for Arlo is he's old now. What is he? He's late 60s or something like that. And he hasn't got it, so he probably is safe. He did not inherit the dominant allele, which I'm sure was a great relief to his kids here. Yes?
Are late onset diseases often dominant alleles?
Not necessarily. No, they can be just about anything. Okay. Yeah. And we don't understand the mechanism of the late onset. So who knows? There's been a lot of research done. You know, the fact that a famous folk singer died of this ghastly disease has fueled a lot of donations to charities that are researching it. So there are Huntington's disease research foundations out there that are working on figuring out what happened here. Okay, a few of the things we know about this disease that's also kind of horrible. It exhibits a property called genetic anticipation. What that means is in each subsequent generation, it gets worse and worse. Yeah, so the idea is, well, if Woody Guthrie had it really bad, and he died in the 1960s, I think he was 50-something, somewhere around 50 or 60 years old, but it hit him at 40. If he had it that bad, if any of his kids had it, they would get it even worse. And we do know a little bit about the mechanism behind that. This is caused by something called the trinucleotide repeat. So inside the gene responsible for Huntington's disease, there's a CAG, 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 CAG. Just a sequence of 3 nucleotides repeated over and over again. And that's variable. And somehow, every time it gets replicated, there's a chance it will add another CAG in there. So the CAG repeats get longer and longer for each generation, and that makes the disease worse in each generation. See, I told you, genetics is going to start getting complicated here. So, yeah, again, this is something Mendel could not even imagine, but it was there. All right, so that's our Guthrie family. Here's another pedigree. This is a famous one, too. So who has heard of Alexis of Russia? Nicholas and Alexandria. Nicholas was the Tsar of Russia at the time of World War I. He was not a very good ruler, apparently. It was under his regime that we had the communist revolution in Russia that overthrew him. He and his family were eventually shuttled off to a prison and later dragged out and shot and their bodies thrown into a mine. Oh, yeah, another terrible end. One of the things that affected his history was this, that one of his sons, Alexis here, had hemophilia. Now, I'm sure you've all heard of hemophilia, right? Yeah, hemophilia is a disease of the blood clotting pathway. Individuals who have this, again, it's a recessive trait. If you have 2 copies of this defective blood clotting gene, your blood doesn't clot appropriately. It can cause a lot of misery and pain. That, you know, we wander around cluelessly on the earth, and every once in a while we bump into things and we get bruises, right? What happens then? Well, the bruise goes away in a day or 2. If Alexis bumped into something, his bruise would blossom into a big blood spot under the skin. It was life-threatening. If he injured himself more overtly and cut himself, he could bleed to death. This caused a lot of anxiety in the Russian royal family. Again, if you read the history of World War I, there's a book called Nicholas in Alexandria about all this. There was a movie, The King's Man. Did anyone see this? No? Oh, guess it's going to bomb at the box office then. Oh, anyway, it's been the subject of movies and so forth. One of the other factors here is that Alexandria fell under the spell of a mad Russian monk named Rasputin, who told them all kinds of stories about how he could cure his son and, yeah, all this stuff. And that was one of the other factors that led to his downfall. So one question is, where did this disease come from? Are there other kids in the royal family who are more likely to be affected? So we do a pedigree. There's Alexis. He's got the trait. He's a hemophiliac. Because it's recessive, we can sort of figure, maybe his mother or his father was also a carrier. Once again, this is going to turn out to be more complicated than just the standard Mendelian recessive and dominant. But we're pretty sure that his mother was a carrier. His mother's mother was Alice over here. And Alice was the daughter of Queen Victoria. Now, you've all heard of her, right? Yeah. 19th century monarch of the United Kingdom, etc. Well, there's Queen Victoria. We suspect that she was a carrier. Because not only was this lineage affected, as you can see over here, there were other European royal families that were similarly affected. So what was, I can't remember where Leopold was. Prussia or something? Who knows? Anyway, we got this lineage over here. This son is affected. His grandson is affected. Over here, this is the Spanish royal family. And in their background, they've got a couple of hemophiliacs here. And those all go down to Juan Carlos of Spain. So we can use this kind of pedigree and trace the history of this. And that's because we've got these multiple instances. That's why we think Queen Victoria was affected with this particular allele. Her parents were not, nor her grandparents, which means Queen Victoria was a mutant. There ought to be a science fiction story about this. Anyway, there's Queen Victoria. She's a mutant for this particular trait. She passes it on to a bunch of her children. She married off her children to all the royal houses of Europe. So she sowed this disease throughout the European aristocracy. Pretty cool trick. But notice over here, this is the British royal family. No, she somehow managed to keep that hemophiliac allele out of the British royal family. And by tracing the sewage. And there's no chance that any of these people there's Prince Edward, Prince Andrew, there's Prince Charles, there's Princess Anne. None of them have it, and they probably do not have any chance of inheriting it or passing it on to their offspring. So another premise for our science fiction novel: Queen Victoria was a mutant who intentionally afflicted a genetic disease in all the other royal families and left her country free of it. Ooh. Man, conspiracy theorists could do something with this. Okay, so that's an interesting pedigree. Here's another pedigree. This is a pedigree composed by this guy, Francis Galton. Remember we mentioned him before? Darwin's cousin, the one who did that silver, gray, and black rabbit transfusion experiment, all this other stuff. The other thing Galton was famous for was promoting eugenics. So he would go everywhere and talk about improving the race and judging people. I always think of him as a creepy, gross, horrible man. Because one of the things he did is he had a little stub of a pencil that he kept in his pocket. And as he walked through the country, as he visited all these other cities, he would make marks for all the beautiful women he saw. So he was actually tallying up who was most attractive. And he actually published a map showing the distribution of beauty in the United Kingdom. And the most beautiful women in all of England and Scotland and Ireland were found in the south of England, where he was from. Look at that beautiful man. So he's judging all this, but we're not going to judge him, right? This also is a very interesting photo. This was, he visited a lab, a criminology lab, that was studying facial and cranial features to see if they could identify criminals. This was a big thing once upon a time, is physiognomy. Let's figure out what the criminal face looks like so we can spot them in a crowd. Looking at this impartially, what do you think? Criminal or not?
Criminal.
Again, it's a discredited idea. You can't do that. But he bought into it. Yes, so he thought. He's a what?
He's a criminal.
Why do you say that?
He looks like one.
Oh, just because he looks like one. Yeah, there's way too many people that think that way. Okay, so he's judging people on their basis of all kinds of characteristics. He's arguing that fairly abstract sorts of things get passed on from generation to generation, like poverty is a heritable trait. It sort of is, isn't it? But not in a biological way. Over here we see a pedigree. Just looking at this again, objectively, what do you think what do you think that pedigree shows? What's the pattern of inheritance in this family over here?
Beautiful.
What's that?
How beautiful they are.
Oh, okay. We'll just roll with that. Say, okay, maybe he's judging this family for how beautiful they are. But the question is, what's the pattern of inheritance? Is it dominant? Is it recessive? Incomplete dominance. Oh, you've been reading ahead in the book. Good for you. So maybe incomplete dominance. Is there any other attributes of this particular family tree that stand out for you?
It only affects males.
It only affects males. Oh, isn't that interesting? Why would that be? Well, here's what it actually is. This is a pedigree of his own family. And what it's supposed to be is a pedigree of scientific ability.
See, I told you it was something. He said brilliant or brilliant.
Yes, he's brilliant and he's beautiful, right? Is that what we're saying? Yeah, so there he is in the pedigree, sort of self-judging himself as a brilliant scientist. And he's saying, okay, my dad was also had scientific ability, and my grandfather. Here's Charles Darwin and his family tree. Oh, man. So here's one of the ways this genetic information gets misused, right? Because, all right, Huntington's disease, yeah, you can see that, you can map it. It's got a very strong overt effect. It's kind of easily distinguishable. There are molecular tests for it, all kinds of things you can do with that. Scientific ability, though, how do you test that? How do you measure it? What are all the factors that go into that? So this is one of the ways Mendel is going to get abused. I can't believe that Mendel would endorse this kind of thing, but who knows? We don't have as much of a record of his personal views. But this is a chronic problem throughout the 20th and into the 21st century, where people look at these subjective things like scientific ability or IQ or poverty or criminality, and we try to hang genes on them. No, it just doesn't work. Okay. That's because there are all kinds of complications in this pattern of inheritance. So I mentioned a few here. So there are a lot of genes. Look at those numbers. 10,000 genes in Drosophila, about 20,000 in us. So, you're trying to figure out what is affecting what. How many genes like that contribute to scientific ability, for instance? It's really hard to sort out. Also, what about the effect of the environment? Yeah, we've got all these people who have been told for generations that no, you're not a good scientist. And it's largely because of environment. Notice that in Galton's Pedigree, no women were identified that way. That's because women weren't allowed to go to college. They didn't have an opportunity during his lifetime. So we got all these environmental influences that complicate it. Furthermore, all this stuff. Yeah, we learn, okay, dominant, recessive. That's how we think of everything. That's not accurate. There's lots of different kinds of relationships between alleles. There's incomplete dominance, codominance, and overdominance. So we're going to see some examples of things that look like blending inheritance. Later, we'll talk about linkage and sex linkage. That's a whole other topic. That's going to get hairy. Trust me on that. It's one of the more complicated topics we're going to deal with here. There's epistasis. So, where was it? Epistasis? We're doing an experiment in epistasis in the lab, right? So we're looking at how eye color is affected. There are all these other things, isoalleles and pseudoalleles. There are some alleles where they look identical to another allele, but they're actually not when you look at a molecular level. So you need new tests to distinguish them. Penetrance is another important one. You could have a particular trait, a particular genetic trait that you've inherited, but it may not be strongly expressed in your phenotype. It all gets very blurry. So several years ago, for instance, Steven Pinker wrote this opinion piece because he had just gone in for one of these newfangled genetic tests where they sequence your genome, and they came back with all this information that said you are prone to baldness, that you're likely to turn bald. And I don't know if you've ever seen a picture of Steven Pinker, but he looks like a dandelion. He's got hair all over the place coming out of his head. So he had to say, that's clearly not the case. What's going on here? And the thing is that there are multiple factors that affect the expression of genes. And here's another concept we'll come back to, pleiotropy. What does pleiotropy mean? What pleiotropy means is that a gene has multiple effects. You know, if Mendel had looked at his data, he would have said, okay, I got this allele that causes yellow peas versus green peas, and that's kind of it, we're done. But no, you have to look deeper, and what you find is that allele may affect multiple factors in the organism. It can get messy. Okay. Here's another thing we're going to run into in short order, and that is the nomenclature. Okay, we've had some slight premonitions of what's going to come down here. Because, I start off, I'm showing you, big A, big B, little b, big C, little, etc. Genes aren't named that way. And then we had an example where I showed you, okay, well, this gene's got a 2-letter name. That's vestigial, remember? It's VG. So we can't use just one letter there. Again, flies, we got 10,000 genes. We have 60,000 alleles, at least, to catalog in Drosophila. So there's no way we can do it with single letters. So geneticists have a bunch of conventions for how we name genes and alleles. And here's the sad news. Everyone's got a different convention. It's infuriating when you read the literature because different species have their names identified in slightly different ways. So we've gone beyond single letter, uppercase and lowercase. So in Drosophila, what we got, we name a gene after the mutant form. So for example, vestigial. Mutant vestigial, that's what it's named after. The wild type dominant form is still called vestigial, but it doesn't cause the trait. In humans, we got another example of this. You've all heard of BRCA1. The breast cancer gene. The breast cancer gene does not cause cancer. Not in healthy wild type individuals. It's the mutant of the gene that causes cancer. So, of course, the cancer gets the credit. It gets named for that. But yeah, BRCA1 is actually a preventive gene. It helps protect you from cancer. Okay, so we're going to name everything after the mutant form. We're going to have a short abbreviation for it. Vestigial becomes VG, for instance. If the mutant is dominant, the first letter is capitalized. Hey, that sounds familiar, right? Otherwise, it's all in lowercase. And different alleles are specified by a superscript with a plus superscript indicating the wild type. What does that all mean? That means if you go to a catalog of fly mutants, what you find is, for instance, VG, you'll find VG in there. And then after it, there will be a superscript with a bunch of numbers and letters after it to indicate which allele we're looking at. Because you can have multiple alleles for a single gene. So the example I'll give here is the white gene. It's named after the mutant phenotype. It's abbreviated W, little w, because it's recessive. And the wild type form is W plus. Put a plus after it. So check those superscripts in problems. And I worked on zebrafish for a long time, and they do something a little different, because everyone had to do it different. I was around when they had the genetics convention where they sort of name what things are called. So in this case, it's all lowercase. You don't use uppercase letters when you name alleles of zebrafish genes. Instead, what you do, if it's a dominant allele, you put a superscript little d. And then often a bunch of letters and numbers to indicate which one. It gets complicated, doesn't it? So you sit there reading the literature, and if you're familiar with the fly literature, you think, okay, I see this name in the zebrafish literature, and it's all lowercase, it must be a recessive allele. No, not necessarily. Got to look at the superscript. And then humans, of course, humans are so obnoxious. They have another set of conventions. All human genes are identified with only uppercase letters. So if you write up BRCA1, you got to write capital B, capital R, capital C, capital A. Not lowercase. It's all uppercase every time. Which means when you read the human genetic literature, it looks like they're shouting at you all the time, right? So all uppercase, then they also have superscripts and all this other stuff. We will get into it more with the lab next week, because next week I'm going to introduce you to this flybase. It's an incredibly useful database of all the different fly mutations and their alleles. Yeah, it's a fun browse. If you're into fly genes, that is. So we got that. One other thing I'll mention about the nomenclature is that there has been a little bit of a pushback, because most of the early work on these genes was done in flies, and fly geneticists kind of went crazy with the names. Yeah, because they're trying to say, yeah, this is just a random gene. We'll give it whatever name I feel like. So yeah, there's a gene called sonic hedgehog identified in flies. You've probably heard of Sonic Hedgehog, the game, but have you heard of Sonic Hedgehog, the gene? Oh yeah, good. It's a really powerful gene. That got a lot of use. So there's a lot of casual, funny names from the fly literature, and then we go to the human literature, and because we're homologous and we share many genes, all these very serious doctors of human medicine are looking up their genes on the databases, and they say, oh, this gene I have just identified to play a role in, for instance, schizophrenia in humans was named lunatic fringe by the fly biologists. And now I've got to go tell my patient that they've got a defective gene called lunatic fringe, and that's why they're crazy. No, they're not crazy. No, doctors don't do that. But you get the idea. So there's been a fair amount of pushback. And what that means is now there's been some proliferation of new names for old genes. Yeah. It would be really cruel to make you read the contemporary genetics literature, because there's got so much of this stuff in it. Okay. So just be prepared for that. There's all kinds of different ways to name a gene. Now, what is affected in these studies of real genetics? Mendelian genetics is kind of simplified cartoon genetics. Real genetics, we've got to deal with some other things. One is multiple alleles. So Mendel, when he looked at each of his traits, had 2 alleles, right? Exactly 2. Very convenient. Because every diploid organism has 2 copies of each gene. Real genes can have hundreds, even thousands of different alleles, different variations in the structure of the gene. So flies, there are some genes that have hundreds of different alleles, and they're all associated with a gene. For instance, white. You can go to the white gene locus, and they'll say, okay, here's a long list. Yeah, seriously, when we go to flybase, you'll see it just dump this long list of all these different alleles, all named W, and weird little superscript with numbers and letters in it. So we get that complication to deal with. And of course, as I remind you here, even if you've got a gene that has 100 different alleles, in any one individual, any one normal diploid individual, you're only going to have 2 possible alleles. And when you're studying multiple alleles, you typically have to study populations or more complicated relationships. And I think you can all think of one great example of that in human populations, the ABO blood group, right? So you got type A, you got type B. Those are 2 different alleles, but you also got type O which is a different allele. So we've got multiple combinations we have to deal with when talking about blood types. Okay, so that's one factor. Another factor is multigenicity. Most traits are regulated by more than one locus. Again, this really complicates matters a lot. So, think of any trait you've got, and in general, it's going to be controlled by multiple genes. It's lots of genes working together. For example, your eye. Your eye is not the product of a single gene. There are thousands of genes working together to make an eye. And this is true for most of the attributes we look at. We've got kind of a misguided view of it all because in genetics we often pick the easy ones to study. Huntington's disease jumps out at you, right? It's got a great big defect in it. Causes a major phenotypic effect. But if you think about it, most of the genetics you have inherited from your family are subtle and small. Like the shape of your face, the shape of your nose. How many amylase genes you have. Most of us probably don't know how many amylase genes we have. Lots of little things like that add up to make the totality of the individual. So most things are multigenic. Another factor that's related to that is pleiotropy. So a pleiotropic gene simultaneously affects many characters. And this again is the rule. It's not a weird exception. Most gene differences, most single gene differences don't affect one single trait in the individual. They affect a whole constellation of traits. Okay, I think I got it. Yeah, here's my example. This is one you've heard of, sickle cell. So sickle cell, the sickle cell allele causes a misfolding of the hemoglobin molecule, which causes them to be prone to crystallize, and then the red blood cells become sickle-shaped, and these are all the phenotypic effects that has. So you get blood cells breaking down, clumping, you get accumulation of cells, that leads to physical weakness, anemia, heart failure, so you get the idea. So sickle cell anemia doesn't just cause, for instance, rheumatism. It causes all of these things. And that's a gene with a pretty large effect on a single cell type. It also has this fascinating pleiotropic effect that you've heard of. It also confers resistance to the malaria parasite. So again, that is a pleiotropic effect from a gene. Anyway, so you got this pleiotropic effect that this gene, which we would love to have, giving you resistance to malaria, but it has this other effect where it causes all these deformities in your red blood cells. So that's pleiotropy for you. Okay, how deep am I going to get into this? We'll see. Okay, I can at least talk about this. So I used this as an example before. This is incomplete dominance. Incomplete dominance causes a kind of blending of characters. So you can see it in things like color. So here's our red snapdragon, our white snapdragon, and if you cross those together, you get a pink snapdragon, which Darwin would have loved. It would have fit with his model so nicely. So we see this kind of blending inheritance. So here's another illustration of this. Well, we can carry this on for generation after generation. So here's crossing a red and a white, we get pink. Then if you cross pink by pink, look what you get. You get 4 different types. 50% are heterozygous, 25% are homozygous for the red form, and 25% are homozygous for the white form. You've seen that before, haven't you? That 3-to-1 ratio in a monohybrid Mendelian cross. Only in this case, the heterozygotes are visibly different from the homozygotes. So that's kind of sweet. That fits with what we know. It's kind of an exception, though. I'm just going to introduce this, and we'll finish this up on Monday, because blood groups are a whole huge another category of things. But again, you're also familiar with this, that blood cells have typically carbohydrates and proteins on their cell surface that antibodies can recognize. And if you get blood transfused into you from an individual who has a slightly different blood type, that your antibodies may recognize that as a foreign substance and attack it. And what it then causes is something called agglutination. That's how it defends itself. The body defends itself. It glues all these invaders together into a mass, which is great if it's bacteria. Get a bunch of bacteria in there, it'll do the same thing. Antibodies will bind to it, it'll agglutinate, then various macrophages and things like that will come along and gobble it up. In this case, though, these are your own red blood cells. So they'll be agglutinated, which leads to bursting of the cells, and that has all kinds of other problems. If you received a massive transfusion, that's a whole bunch of exploding blood cells in your circulatory system, and that can lead to clumping the blood, and then you have serious problems. You don't want that. So that's why you go in for blood typing before you receive a transfusion. They want to make sure they're getting the right kind of you. Now, what's really complicated about this, and we'll get into on Monday, is there are at least 33 different carbohydrates and proteins on the surface of the blood cells that differ in different segments of the population. So ABO is one. There's a couple dozen others that can also affect this. But we'll talk about why they don't kill you if you get a transfusion from somebody with the MN blood type and your type N or whatever. That's on Monday. Okay, any questions before I let you go? Remember, you're supposed to ask me questions as we go.